Professor of Agriculture at University of Western Australia, very eminent scientist in the area of agriculture, and he has been a great friend of ICRISAT for several years, about more than I think 25 years or 30 years or so. And we always enjoy his guidance, his collaboration with ICRISAT. So we are grateful to you, sir, for coming to ICRISAT and for accepting our invitation. So you can see that house is full, and all of them, they are very excited to or looking forward to have your presentation. So for some new colleagues, so I think Dr. Siddique name is not new for many of you, but for new colleagues who are just starting PhD or so, let me give brief introduction about Professor Siddique. As I mentioned that he is having 30 years agricultural research, teaching and management experience. Jonna, you can come in the center, please, because please. So yeah, so he is having 30 years experience in agricultural research, teaching and management in both Australia and overseas. He has developed a national and international recognition in the agricultural science, especially in the fields of crop physiology, production, agronomy, farming system, genetic resources, breeding research in cereal, grain and pasture legumes and oilseed crops. Professor Siddique's publications are considered as key papers in these fields, and they are widely cited. These publications includes numerous highly cited papers on adaptation, physiology, and genetics of crops to dryland environments. Professor Siddique has conducted research on adaptation of crops to water deficits and the phenological, morphological, physiological, biochemical, and genetic traits that enable crops to cope with various abiotic stress. As a result, Professor Siddique's personal research and with others with whom he collaborates, Australia has become one of the major grain legume exporting nations in the world. In fact, now you know that Australia is probably number one chickpea exporter to India, and Professor Siddique has played key role in enhancing or in fact starting from the introduction of chickpea in Australia. So, and he has received several awards for that one. And right now, his pioneer research on chickpea has contributed enormously to Australian chickpea industry, which is currently valued at more than $300 million per annum. In 2015, Professor Siddique was elected as the International Fellow of the Indian Society of Plant Physiology and the Foreign Fellow of the Indian National Academy of Agricultural Sciences. Professor Siddique is the winner of the Western Australian Year of the Award 2014 for his contribution to agricultural sciences and farming community. Professor Siddique has published more than 250 scientific papers, book chapters, and he has published several books, technical bulletins, and he is basically editorial board member of a range of journals. And as I mentioned earlier, on top of that, he is a well-wisher, he is a good supporter for our research at ICRISAT. He has also been guiding CRP Grain Legumes program by serving as a member of International Advisory Board. And he has been collaborating with us for many, many years. And right now we are having one important project, Australian Indo-Scientific Science and Research project, AISRF project. So with all these things, once again, we would like to welcome Professor Siddique for this lecture, and please join me in welcoming. And we would like to welcome our Director General, Dr. David Warwickson, Joanna, and many other senior colleagues. Hall is full. So this indicates that how much people are keen to listen to you, Professor.
Good morning, and thank you, Rajiv, for that lengthy introduction. And it's always a pleasure to come to ICRISAT. And in fact, 1991, I gave a lecture in this seminar room that the theme or the title of the seminar was chickpea idiotypes. Those days, Chris Johansson was the director of the grain legume program. And of course, over the years, we have built up enormous collaboration with ICRISAT, trained a number of PhD students, and I look forward continuing that with ICRISAT and the University of Western Australia. Many of my colleagues has contributed as well in this collaboration. It's also good to see a lot of young people in the audience, which is again a good sign, David, of the future of ICRISAT. So I just put a question: can we breed for efficient root system for water and nutrient uptake in crops? Why I chose this topic today is because so much we have spent on the above ground, and we haven't spent that much in below ground. And the reason is very simple. It's so difficult to quantify, so difficult to phenotype, so difficult for the breeder, and the G by E and management interaction is going to be significant. So that's going to be a difficult topic. But if you look at the future, how are we going to improve crop productivity, especially with the climate change, nutrient acquisition efficiency, and so on? We have to do something on roots. So I tell my students and others, if some of you work on roots related topic, whether it is genetics, agronomy, or physiology, it will contribute to the root science. So in our university, the institute has got a theme called crop root rhizosphere, and we are going to give a lot of emphasis in that. In that process, we would like to have partnership with ICRISAT in a variety of areas. So I just would like to acknowledge the Australian Research Council, which is quite a prestigious funding organization, Grain Research and Development Corporation, University of Western Australia. I should also acknowledge the CRP Grain Legume Program, SHOBA. There has been some funding from ICADA to phenotype the chickpea roots. I'll show one slide. It's currently being written up. Now, those who have not been to University of Western Australia, this is the city of Perth, the wonderful Kings Park, about 1,500 hectares. This is really interesting. And this is where the University of Western Australia, wonderful place. And those who haven't been, including David, please do visit us. So this is so the Perth is the capital of Western Australia. Western Australia is almost more than half, it would probably 3/4 of Australia size, a population of about 2.6 million. That's about 10% of Australia's population. Majority of those people are living in Perth, 1.6 million. And if you look at the largest city in the world, the entire population living on the coastal area. So I live very close to the Swan River. So this is the university, wonderful university, and my department and institute is here. This is where the hub of the Harvey Miller's Center, Center for Plant Energy Biology, and so on. So a Mediterranean style, more like Stanford University, 103 years old, the university. We are a member of the Group 8 universities. This is very important. The ranking, universities are now really looking for ranking. The ranking we follow is the academic ranking of the world universities, formerly called Shanghai Jiaotong. So in 2015, overall we are 4. Overall means all categories. And in the world ranking, we are 87. So for example, if I gave a talk about 15 years ago, we would have been somewhere around 500. When we got the Nobel Prize in Medicine in 2005 for the Helicobacter pylori, the stomach ulcer, then we jumped up to 200. Then our then Vice Chancellor said, we must start moving forward. We should not look within Australia. We should look internationally. How can we move forward? So the strategies work, the culture changed, the behavior of the people, the staff and students changed, and we started moving upwards. Of course, this is one indicator. There are other indicators in the university system. But the most important thing is that in life and agriculture science, we're number 1 in Australia. That is overall agriculture. We have plants, crops, animals, soils, ag economics, and so on. In 25 in the world, just behind University California Davis, which is really good because that's a larger institution than us. But there's another ranking we follow. In agriculture alone, you take out the plant or life science out, we are 30. In plant and animal science, we are number 18. So the university has put a benchmark by 2050, we want to reach 50 in the world, which is an aspirational goal. The ranking may change, but that's good. Everyone in the university, from finance people to the professors, they all behave like that. So you need to have an overall goal. Where do you want to be? It could be impact on the rural livelihood of people for ICRISAT. And that's the type of a message the hierarchy has given us, and we have changed our behavior and culture. So the outline of my presentation is, I'll give an introduction, the climate change and abiotic stress, importance of crop root system, a search for efficient root system, genotypic variability, phenotypic plasticity, and foraging for water and nutrients, and draw a conclusion and leave a lot of time for discussion. Climate change and abiotic stress. So I just need to just give a brief background. Climate change will add substantial pressure to the existing system. In relation to Australian agriculture, terminal drought is very common. Acidity linked to the aluminium toxicity. So a lot of the breeding programs are geared for that. If we don't overcome that, we cannot grow crops like wheat or legumes. Soil compaction, again, the deep tillage, and that sort of systems is going on. Alkalinity, boron toxicity, and of course, inherent low fertility. Australia is the driest human inhabited continent in the world. We should not do agriculture, but we are doing it with that fragile environment following the principles of conservation agriculture. Drying and variable climate, water is the major limitation, and of course, that is then again soil situation increases the stress. So you can see here the scenario for 2030, temperature is increasing, rainfall is declining, and evaporation is increasing. I'll come a little bit more details to that. So this is where the grain belt of Australia. So a lot of Australia is useless in terms of agriculture, but if you look at this, is where majority of the agriculture is grown. So 300 to 600 millimeter rainfall ISO heights. So this is where I'm coming from. So we have a Mediterranean type climate, and that extends a little bit there. Then here it is more like the northern Indian situation. So the climate is cold, wet winters and hot, dry summers. About 12 million hectares of cropping in this part of Western Australia, 4,000 farmers. So 10 years ago, we had 14,000 farmers. So now the farmer number has declined, but the farm size has increased. So consolidation and market failure, and people then moved out of agriculture. Efficient farmers are coming into agriculture. So 3,000 hectares is the average farm size, but we have farm as big as 100,000 hectares in Western Australia. So a lot of the marginal areas, the farm size is 10 to 20,000 hectares. Wheat, barley, canola, grain, legume, and pasture and sheep in this part, but quite a lot of cattle in the northern part. So we have divided our grain belt into low rainfall, 250 to 325, 325 to 450, then what we call the 450 to 750, then the very high rainfall. So majority of the agriculture is practiced in this area. So the season gets shorter, warmer here. The season gets cooler and longer here. The rainfall declines as you move to this way. So this is a snapshot of 1900 to 2009. Now it's about 30% reduction in rainfall, 10% here. In the Queensland, that's the area, 8%. But you are seeing an increasing trend in rainfall in the northern part. Now let's look at the actual rainfall. So I just said we are drying, but this graph shows that we are getting wetter. That is the total Western Australia, not the southwest of Western Australia. So we're getting some increasing summer rainfall in the whole of Western Australia. But let's focus on the agriculture area, the southwestern Australia. There is clearly a trend of declining. We have seen significant drop in rainfall in the southwest since the mid-70s. And in fact, the driest since 2010, low variability, and 2010 is the driest on the record. So we're really seeing significant impact of climate change in relation to rainfall in the southwest of Western Australia. Let us look at the summer. There is no trend. Autumn, yes, there is a decline. Winter, there is a decline. And spring. So you need to look at the distribution of the rainfall, not the total rainfall alone, and what impact it has on agriculture. How does the crops and cropping system survive? So if you look at the spatial distribution, again, it is getting drier here. This bit is getting wetter, and the government has embarked a big program to have tropical agriculture, including parts of Northern Territory and Queensland. And we're going to establish a cooperative research center very soon with several $1 million, up to $25 million per annum for the next 7 years to look at a different kind of agriculture for the tropical areas. There may be opportunities for ICRISAT to collaborate. I'm sure Peter will work on that. But if you look at the Perth Dam flow, this is clear indication of how we are drying. The Perth Dam flow, this is sitting around 320 gigaliters per annum. And since 1975, stepwise reduction, we have really going something like 50 gigaliters into the Perth dams. Again, so those people who doesn't believe in climate change, this is just a good example. We have measured that. But as a result of that, the government of Western Australia has started 2 desalination plants. And also the culture of the people have changed. We used to water all our garden and so on. Now we're using only half what we used to use before. So the message has gone, and the taxation has gone into water consumption, so people's behavior have changed. So we learned how to manage drought, and the Californians are coming to Australia to learn, particularly to Western Australia, how do we adapt to climate change and the city people. It's not only the rainfall, but if you look at the temperature, which is another driver of climate change, really we can see that southwest of Western Australia, or Western Australia has increased the temperature to 13, the hottest year on record. I don't know what's happening this year, but perhaps it will be still hotter again this summer. But let us look at the southwest of Western Australia. That was the whole of Australia. But again here, 4 hottest years in record. Look at that. So improvement in heat tolerance in crop is going to be quite important in Australia, particularly crops like wheat and grain legumes. So hence more stress on heat tolerance, particularly around flowering reproductive stage. So importance of crop root system. I think this is quite a conceptual framework. I just want to quickly go through basically what is the major driver for production in the drylands. So you got the soil water at sowing, whatever is stored in the soil, growing season, precipitation or rainfall, that decides the water supply. Of course, you lose from runoff, deep drainage and soil evaporation. You need to reduce that, and water not extracted by the crop, either because massive clay structure or the root system cannot go there. So we got scope for improvement. So water transpired and the transpiration efficiency, again, I won't go into the physiology of that, the biomass production, partitioning harvest index, grain number, grain weight, besides the yield. So we can do a lot of things in order to adjust this and improve that for the dryland system. But I think the main thing is root system, the fundamental to crop productivity. And in order to overcome some of the abiotic constraints and maintaining the structure and function of the properties, root system is very important. Important target in breeding program for rain-fed environments. I don't know many breeding programs really targeting that because of the difficulties associated with phenotyping and genotyping until now. So the root traits contribute to enhanced water nutrient. One of them is the deeper root system. The other one is increased root density in the subsoil. Increased root hair length. I'm just listing some of these things. And you can also play around with the xylem diameter, whether we can increase or decrease, and that will have impact on the hydraulic conductivity, how the water is then metered and used. So there is a lot of fundamental scope where we can look. This is just an old study when I did historical wheat varieties. So there was something like 20 or 30 varieties. But I just wanted to show one piece of data. This is the first one of the first varieties released in Australia, wheat 1860s. At that time, I chose another one, 1960, 61, 79, and 86. And you can see that the breeders have reduced the time to flowering and also to some extent physiological maturity. We had at that time some RHT gene, no gene, 1 dose of RHT, 2 doses of RHT, and I also included a barley cultivar. But I will just show one data. So here, the root dry matter. Here it is the shoot dry matter. I forgot to label last night. This is actually the modern variety Culin, and this is the purple straw. This is the Gamania. Basically, this is the one-to-one line. And what you can see that in a modern variety, it started partitioning more towards the shoot much earlier. So in other words, we have reduced the root system by breeding because we were looking for harvest index improvement. So what it implies is that you don't need a very large root system and invest a lot of biomass in that particular environment. Why not that partition that energy or carbohydrates or photosynthase into the top? With the RHT 0 and 1, there was no difference. So we have already reduced indirectly some of the root system unknowingly, but we haven't really targeted a breeding program in order to cope up with nutrient and water uptake. So I'll leave that old data there. Now I'll move into some of the work which we are doing. Basically, the productivity by selecting root traits, root architecture is important, genotypic plasticity, and foraging strategies. So it's not just about the root structure. There are strategies how which you can take the phosphorus and nitrogen from the soil. But the problem has been phenotyping, which is really the biggest hurdle. No matter whatever we do, we have to go through this process. And combined with the genotyping, we got hope to do something in the root work for the next 10 years. So we developed what we call a rubbish bin technique or semi-hydroponics technique. This is the backyard bin, 1 meter tall bins. And these are the pouches, which is a waterproof black plastic pouches. And there is a pump here, which circulates the water and nutrients. So it's not a completely hydroponic system. This is a lupin plant germinated, and they were then kept on this one with a clip, and the root system grows nicely under the dark. We can occasionally open and use the camera. When it hits the bottom, we terminate the experiment. So from there, we can then work out the true genetic potential, and we screened the entire core collection of lupins, which is about 125. But you get really good things like the first order branch, second order branch, the tap roots, and also the root hairs, including the proteoid roots. And this straight into the computer system where you can then generate into quantitative data, and coupled with the genomic data, then we will get some true genetic potential and root architecture. So searching for efficient root system, genotypic variability, I'll show some data. So we looked at something like 35 traits. Some of them are derived traits, some of them are primary traits. But here it is the minimum and the maximum value. And you can see tremendous variability in root length, root surface area, top 20 centimeter layer root length, 20 centimeter layer branch length, and the total root dry mass. That's from that work. So we used here in the case 112, and that represents the world collection of about 2,000 accessions held in the gene bank. I'll show one data. This is the lupin genotypes, and this is the taproot length in millimeters. And 2 weeks in that system, already variability is existing. By 6 weeks, wonderful variation. It's very clear, repeatable, and no effect of other environment. That's the true potential we were able to measure. Let us look at another parameter, number of first order branches per plant. So again, genotype 92 to whatever genotype. So tremendous variability, we were able to look at it. Once we got this information, the question is that, of course, I'll come back a couple of other slides. How are we going to do this in different environments? Fortunately, our group has developed the root map model. Anthony, you probably know some of these guys, and SimRoot models, really based on some very good mathematical and sound biological principles. So here is I've shown 4 genotypes, 1, 2, 3, and 4. These are actually measured by the semi-hydronic system. This is simulated by the root 2 models, which gave us confidence that we can then go and simulate such system for any environment and then look at what is the probability. Because I don't want to start a breeding program, invest 10, 20 years of money into that, unless we are really clear that it will have an impact. Will it take up extra phosphorus or nutrition, or will it take up that 20 millimeters of water towards the end of grain filling? Unless you have got some real feeling, don't get into that sort of breeding program because it is going to be difficult. So we're getting into that direction. So root depth, branching, root density at depth. And these are all published. Anyone interested, I'm happy to provide the copies. It's not only us. Manshadi et al. In 2008, he looked at the number of seminal roots in wheat genotypes. Remember, we have got 2 types of root system, the seminal and the nodal root system. The nodal one comes from the tillers. The seminal is what we call the taproot. And here we have a number of Australian wheat varieties, plus also some of the CIMET ones. So tremendous variation in the number of seminal roots per plant. I'll only show one data on the chickpea. So we have just finished phenotyping 275 accessions of the chickpea, which include 250 the core collection or reference collection. The 50 never reached Australia for various reasons. And this data is currently being processed, and the manuscript will be done, and the data will be available for people like Rajiv to look at some of the genomic things. But look at that here. It's a 4-week-old chickpea. This is one genotype. I don't have the number here. And look at the taproot going, whereas look at the type of thing. This will have some effect on water nutrient uptake, and that needs to be tested. but we have got the data just being processed at the moment. There's another work by Manchadi here, again showing the greater root density in the top layer. Hartog is an Australian wheat variety, whereas Siri 82 is from Simet. See, these sort of things will have an enormous effect on nitrogen uptake, because nitrogen is mostly on the top layer. And if those type of root system can take the nitrogen, a lot of the nitrogen will not be leached out of the system. Or some of this root can give the signal when the soil layer dries in the top, and ABA can be transmitted to the stomata, and then you can have the response to stomatal response in relation to drought. Moving on, phenotypic plasticity. Basically, drought is one of the major constraints, whether it is in Australia or in the ICRISAT mandate regions. Terminal drought significantly affects crop yield in Australia. For example, we plant in winter. The spring wheat is planted in winter. The vegetative growth phase is pretty good. Rainfall, low temperature, high humidity, low evaporation. As soon as the plant gets into the booting stage, then high temperature, low humidity, high evaporation, low or no rainfall, leading to terminal drought, yield reduction. This is a clear case, and that is being now increased by the change in climate. So one of my PhD students, she's from India, finished her PhD. She looked at a system where the blue color is missing. This is a split root system where part of the one half is kept from the other half in a watertight thing. So wet, 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 dry, dry, dry from flowering onwards. And we used genotypes known for resistance, dry stale, some of the common genotypes in Western Australia, and the breeders' materials, etc. Basically, the specific root length responded differently to drought. So this sort of system helps us to study what does the root system do and she went and looked at the ABA and stomatal opening and so on. And that may be another story, another day. So coming back to the lupins, we had these 112 genotypes. Then we looked at the contrasting genotypes, so 16, 44, and 85. 23 days, well watered, and the total root length centimeter per plant. So you can see that. Then what happened was we water tested 49 days. Tremendous jump in terms of the plasticity. Whereas this particular genotype, 044, were more stable. So if we didn't have the phenotypic data for the entire collection, we were not able to do experiments like this, because people asked that you got all this data, this was not done in the field, and what are you going to do? The answer is that at least I get the total genotypic variability without any stress. And then that true genotypic variability, then I can use for manipulation studies, either in the field or in the glasshouse studies. This is another example where we took a genotype 71 and genotype 85. There are three systems, nil phosphorus, top dress phosphorus, banded phosphorus. So this is the top dress, and this is the banded. So you can see that genotype 85 responded quite significantly in terms of the top layer root length density, in this case because of that. So these are quite important in terms of managing your root system. Foraging strategies. The majority of the flowering plants have mycorrhiza symbiosis. I think I heard that, David, you are initiating soil biology and microbiology here in ICRISAT again, and we have a strong group at UWA who is in soil biology and related areas, including greenhouse gas emission from the soil system. I was just talking with Anthony this morning. And some of them, like Proteaceae family plants and some of the lupin species, particularly the white lupins, develop cluster roots to adapt. Again, our group, Hans Lambers and others, are very much into this, looking at a low phosphorus status in Australian soil. How does the native plant survive without much phosphorus? Can we learn some of those things and then look for such system in croplands? In fact, we're just going to start the entire chickpea co-collection for phosphorus acquisition efficiency will be commencing within the next 2 months. Look at variability, and again, linking with the genomic data, then we should be able to identify chickpea with high phosphorus acquisition efficiency, especially a lot of the soil-bound phosphate in Indian soils and Australian soils how can we forage that soil-bound phosphorus in a cropping system with the chickpea followed by wheat or rice, so that a lot of those phosphorus can be unlocked. Narrow leaf lupin plants do not form cluster roots and does not develop effective mycorrhiza. So there must be some other mechanism. For example, altering root architecture, which we just saw, increasing acid exudation. This is quite important in case of chickpea and PGNP as well. This is the power of the simulation model. So what we did was, in our experiment with the long tubes, this is in the soil, we localized the phosphorus here, and this is phosphorus here. But our actual study and simulation shows that if the phosphorus is not missed by the root system, you can have an entire root development. So a root system with very narrow, less branching in the top layer, if it missed the banded phosphate, then entire root system can have effect. This sort of information was, again, we were able to generate because we looked at the variability within the accessions. Another data we generated here is the carboxylase. So the major acid is citrate, but citrate, malate, acetate, and the total. So the total is here. So you got the zero phosphorus. This is inert soil, no phosphorus, and you got 1, 2, 3 genotypes. You can see that the major acid secretion is citrate, not much variability there. But as soon as you give a low dose of 50 phosphorus, you can see that genotypes differed. So basically, you need some phosphorus to kickstart, and then the plant triggers the acid excretion, and that will help to scavenge the phosphorus which is locked within the soil. And when we increased more phosphorus, of course, there has been reduction in the secretion of the organic acid. So we now want to look at that with more detail, because we are moving into a stage where the conservation agriculture, we cannot continue to put a lot of phosphorus and nitrogen into the system. We must reduce the input, particularly in the marginal areas, and then save that money for the farmers, but also optimize the production. So these sort of things, through genetic approach, will help us, not just looking at the yield alone. I think this work, again, is quite a nice work from ICRISAT, where they looked at the root volume and then moderate drought and severe drought, and the correlation with seed yield. And you can see that as the drought increased, the correlation is better, whether at different layers or the total layer, which is quite a nice piece of work. Again, you guys took that work into breeding populations, and so-called hotspot. I never understand that. What is that? There's a lot of stuff in there, whether it is drought tolerance or canopy development. I think Vincent showed some of the work he's doing in the scanning work he's doing. So I think these are quite important. If we can get some of these answers sorted out, then we feel confidence into the breeding program. Then ask the breeders to start the crossing. You don't need to go and phenotype everything. Take it to the right target environment, test and see whether they yield, because we will not have the answer for everything, but as long as you can really tag and say that there is something in that particular region and the parents have really been characterized well, so we got some sound scientific basis of moving forward to develop efficient root system in various crops. So I borrowed this from Rajiv's paper. Again, what we are trying is, we got the genetic resources. If you don't have, we go and look it into the germplasm, or create, as I saw today, Srinivasan showed some of the wonderful work in terms of mutation work they are doing in the creating variability. Then the question is that how do we move that into the improved germplasm? People get so much scared when they say all these things. These are all tools. If these tools are not utilized for the breeding program in this instance, then not much use. Okay, good papers are important. This is phenotyping is going to be more critical. David, you showed one of the slides in the Delhi conference. The genotyping, the cost of genotyping is going down, but the cost of phenotyping hasn't gone down. So that's where I think I'm happy that ICRISAT is putting a lot of effort. So as others, we must work closely in phenotyping and targeting the right environment. in order to move from here to here as soon as possible so that we don't need to spend 15 years to develop a chickpea variety or 10, 12 years to develop a chickpea variety. We need to know what exactly we're doing, where we're going, what is our target environment, what are the traits, even if we don't know individually what are the traits we're targeting, then we got really room for improvement rather than a black box approach. So this is my conclusion. So basically, genotype variability provides a promising approach for selecting efficient root system for abiotic stress tolerance. You need to define that. The modification of root system architecture could contribute to improvements in desirable agronomic traits. I showed some examples. I think development in phenotyping, imaging, and molecular marker technologies will help us because there has been people 20 years ago started working on wheat, or 30 years. They didn't make many progress because they were not able to phenotype that in the populations. I think the challenge is that physiological and genetic advances to translate that into the applied program is still a challenge, but some consortium need to be formed, and internationally we should work together such approach so that we can selected crops, we can make rapid advances in root breeding for efficient root system. Thank you.
So thank you very much, Professor Siddique, for very impressive presentation. And we are really, I think, like me, many of us, we are very excited to listen all these advances in that looping breeding program and other examples. So now floor is open for some discussions. Okay, Arvind, please. Arvind is the one who is doing that root trait breeding for rice.
Rice, yes, I know.
Arvind, please.
So we took some of these things into some of the soil types and looked at that, and of course, as you know the germplasm, many of those germplasm doesn't have the yield ability, right? So the next challenge is to make the crosses, but we were able to show either by experimentally or taking to the field whether they were able to take more phosphorus or more water from different layers, but a lot of those are from the genetic resources. They're not having the yield trait. So no, but some of the work done in Australia, for example, the development of dry stale variety, yield advantage of about 5 to 10%. The narrow xylem diameter by John Passiora's hypothesis, Richard got together and developed some varieties which has got advantage in certain situations. Again, I re-emphasize that unless we have the target environment well defined, then we will be in trouble. So a lot of these things we worked on germplasm. We haven't developed breeding populations. Yeah, I think they are all valid comments. I showed the example of the phosphorus and water stress where the genotypes triggered different things, so it will do that. That's a good response. Whereas some genotypes, the one genotype I showed, it did not change at all. So in a situation where there is more water at deeper layers, if the root is not going deeper, you won't get that water. In some situations, the soil depth doesn't allow the roots to go deeper. There's no need for deeper root system. In terms of root cost, the energy cost to have a root system growing, yes, I showed that example of the purple straw versus the Kulin wheat variety, which was done in the field, 6 months in the field. Breeders have indirectly reduced the overall root system. So we need a efficient root system. Then in terms of yield, etc., you need to get the phenology and things right. Sometimes what happens, you will have a large root system, but the flowering time may be much later. So they start investing because the sink is not there into the root system. So all those things we need to look into the context. We cannot change the flowering time at Patancheru. We need that 35 days after sowing flowering time. Then you look at what is the best root system in a black soil here, where there's a lot of stored moisture, either by recharging the profile or through the summer rainfall. Can we get that extra water in order to get the grain filling? The work by Vincent and our work also shows that it's that 10 or 20, in fact even 10, 15 millimeters of that water available at the grain filling period can have a significant impact on water use efficiency. more than 50 kilograms per hectare per millimeter. So they are the type of things we need. And again, I think we are in a situation where we have got some powerful simulation models plus good genetic understanding. We can make some calculated really breeding in this situation. Thank you. Vincent, please.
I just wanted to get back on your data on wheat and the comparison of all varieties versus the new one, where clearly, breeding has reduced the amount of food per unit of fruit. And it's not the same, it's not only true in wheat, it's probably true in many major crops, especially cereals. But I think it's a clear demonstration that what we aim at when we talk about adaptation is not so much the dense and profuse root system, but more efficient hydraulically. So what is a way according to you? For example, you're working lupins. You seem to be again looking for densities, and so what are you specifically looking? Are you looking
I think,
in particular to some hydraulics?
Yeah. So I think there are three things we could look at. For example, we have very sandy soils in Western Australia, so the root can go as deep as 3 to 4 meters, and there is water because not much organic matter. A lot of the rain gets into there, and many crops doesn't have the access to that. In that particular target environment, rooting depth is more important. In terms of the wheat, a lot of the nitrogen is applied on the top, right? And it doesn't go into the deep. And earlier on, if you have a root system which can then take all this nitrogen, clearly that work is very important. Similarly, the top 30 centimeters dries in our environment faster. And if the root can then give the signal to partially close the stomata and then regulate water flow, that's important. The phosphorus uptake, again, you need. So you really need to look at what we are looking in. Deeper layers where there is a lot of water, a deeper root system is needed. So depend upon the target environment. So this is, you're talking about chickpea, irrigated chickpea, right? Yeah. Normally, majority of the chickpea in the world is not grown under irrigation, except in the northwest of Western Australia, where I developed a variety called the Kimberley Large, where we have plenty of water from the Ord River. Then part of the Sonora Valley, Mexico, they also irrigate. I think irrigated chickpea will be different. In that case, you don't need a very deeper layers. You really have an efficient system where the applied water is taken up and the next lot of water comes. But what in an irrigated system, what we need is we probably need between intervals of irrigation the plant to survive a longer time. So we can have the number of irrigations less, but if you continue to apply water, the chickpea will not develop a deeper root system. They will be more like growing plants in a pot. They will be waiting for the next dose of water. I think supplementary irrigation, if you're asking about chickpea, it is practiced in the West Asia and North Africa, the Kabuli large chickpeas where they give supplementary irrigation. We need to look at the critical stages where we have, perhaps around the early podding time so that the seed fill can occur. But I would not recommend, sorry? Yeah. So we really look at a different approach there. You know, what is the critical stage at which the irrigation has to be applied? What is the economic optimum there? And continue to apply a lot of water, that's a waste. Yeah. Yeah. So there is a paper by 19 authors, 16 authors from India and 3 from Australia, which I'm one of the authors, where we looked at, Vincent, I can't remember, probably 45 or 75 genotypes. I think something like 25 or 30 locations in India, as north as Hisar, as south as Gulbarga. Pur and Gaur is one of the authors in that paper. We looked at Jens Berger led that work with an ACR supported one. Basically, yes, you are right. You can have 35 days of flowering. You take it to Hisar, it will be double that. So we really need to look at the photoperiod and temperature response and look at that. The problem is you can have an early flowering in Ludhiana and Hisar, but then the temperature is very cold. So the flowers don't set the pod. They wait for the warmer temperature. So there is a trade-off there. Having said that, if you're looking in the rice fallow system, then some of the varieties developed by ICRISAT program, the super early ones with good quality traits will fit in. We need to look at the cropping system. So for example, the northern belt of India, the chickpea area has declined dramatically. Punjab and Haryana doesn't grow any more chickpeas. because other cash crops or rice and wheat came into thing. So we need to look at where they're fitting. And you got the germplasm here and look at. But basically, it is the photoperiod and temperature driving. And if you take the photoperiod out, then it is the temperature. So it will still be flowering early compared with the photoperiod responsive. But I'm happy to send that paper which deals with this issue.
Okay, very good. So I think that we had really good discussions and okay, we are having, okay, sorry. So one Stefania and then Pallavi, then we will close this. Thanks.
Well, first of all, it's always nice to listen to your presentation, Kadambot. My question is related to, you are talking about looking at different
Of course, as you know that they take, for example, the chickpea reference or co-collection of 300, there's a tremendous above ground variation. But what we looked, same with the lupins, there's below ground. When we take, you've been taken to the field into a population structure, how they, etc. We haven't looked at, but some of those genotypes were taken into the field and looked. There is a paper in Field Crop Research looking at deep tillage and how they respond to do that. I think we need to look at it, but I think it's an early stage where we are at the beginning of this variability. The next stage will be, I don't want to take them into the field and look straight. If we convinced, we need to really start making crosses, and that needs to be then phenotyped in the field. Sure, there will be variation.
Pallavi, yeah.
Under drought conditions, the way root architecture and root morphology changes in the same way we have seen a dramatic change in leaf architecture as well. So they too have gone to any analysis for these root structures.
No, we haven't looked, but we got the photographs and things. I mean, there is a good relationship between, for example, if you have got a chickpea with large leaflets and things, which will eventually lead to larger seed size, they have to invest some of those photosynthate somewhere. I think they will invest in the root system to start with. But as Vincent and I just discussed that, they should not continue to invest. I showed the example of the modern wheat variety. They invested earlier on when there was no sink. Then they straight away started when the sink, in this case the stem and the ear started developing, they partitioned more. So you need to have that strategy. So you could have a chickpea with or groundnut where large leaflets, etc. They may eventually give large seed size. They will invest in the root system, but later on when the sink develops, they must start doing. But in order to develop that sink, you need the nutrients and water. Perhaps that's the strategy. So it's not going to be rigid. It needs to be plastic and dynamic like the above ground. Again, I don't have all the answers, but it would be nice to do the next 10 years of my career to work on roots with some young people.
Okay, last question from Joe. I like those.
I gave a talk in New Delhi that was on wheat, where we looked at the VPD response in relation to VPD and transpiration in 20 wheat genotypes. So the basic hypothesis was the leaf temperature is regulated by transpiration when you cool, so the stomata regulate. But the root can also play a significant role. For example, depending upon the root hydraulic conductivity, one of them, there is no resistance, simply pump the water like a tap, and then you run out of water. There's another one, what will happen, the resistance is built up, and hence the water won't be. And things like leaf rolling, the vaccinus, etc., can also help to conserve that water, provided the condition improves later on, etc. So root is one trait, but there are other traits, stomatal regulation, then vaccinus, leaf rolling, then ABA simulation by root gives a signal. but some of the signals are coming from the root because root is the one who is bringing the water and nutrient. I'll ask the genomics people, so it'll be nice to do that. But there is good correlation between some of the work done in the glasshouse in relation to root and other traits into the field. Again, one need to be very careful. You need to know what environment you are targeting. If you just go and look at a universal drought tolerance, it's not going to happen. But QTL, I don't want to answer on that. Yeah.
So I mean, in the terms of QTL, so if people are generating phenotyping data for plasticity in some crops, and if they have done, then they can identify the QTL. But I don't know that many times or many people, they have done the phenotyping for plasticity. So QTL just business of the phenotype. So probably Arvind wants to say something about that. Okay. Okay, good. Oh yeah. So Arvind, the next question is whether they are sitting in the same area where you had those yield related and these things. So this indicates that very clear. Well, so that basically that means that you are having something there.
I want to make a comment on, we need to be careful when you talk about plasticity, because when by plasticity, first of all, we need to define what we mean by plasticity. And I think in the back of our mind, maybe without fully understanding it, what we talk about or response to environment. Yeah, there are already QTL for what we talk about here, like plasticity. For example, the response of leaf expansion to BPD in maize, in leaf 6 of maize, is probably what you call QTL for plasticity. So it's basically the genotypic difference in the capacity of corns to keep on expanding leaves under a certain threshold of BPD. So that's plasticity, but we need to be careful what we mean.
What is the plasticity?
If we measure things under 3, 4, 5 environment, get QTLs and talk about plasticity QTL, we're still going to be in the dark. So unless we really define what we mean by plasticity, well, we won't make much
I think
progress.
Arvind wants to.
But it's very like, breeding for yield, you cannot have that one environment, there is a plasticity. But what will interesting will be with the STIP-P1 once we go through the data, then if you are convinced with some of the key traits, we need to look at some crossing and develop some populations, and that could be quite interesting. It will take time, and it's not going to be easy for breeding for efficient root system. But we have to start somewhere. Otherwise, we will be again in 10 years' time, we will be talking about root, but we haven't made progress. But people like Jonathan Lynch group in Penn State, they have made good progress in relation to the Phaseolus, common bean, where they have developed a system where efficient phosphorus uptake. I think he's now working on the maize, the corn as well. So perhaps ICRISAT and us get together and work on chickpea root system for the future.
Very good. I think we had very good discussion. So with these words, we would like to thank once again Professor Siddique for this great presentation. And on this occasion, not only for presentation on behalf of ICRISAT, we would like to thank you very much for our collaboration and support and for friendship, what you are extending to ICRISAT for last several years. So we are really grateful to you, and we look forward to have more collaboration with you in coming years. Thank you very much once again. And thanks to all of you for coming and to participate in this seminar. Thank you very much and have a nice day.